I'm going to talk through what we're doing on this broccoli project a little bit historically and a little bit on the current efforts. We've been getting a little bit of notoriety over the course of the past year in the popular press, so it's kind of been on the spot for various audiences. So we'll see how I do with this audience who actually knows the subject matter very well. So overall, we're looking at building an Eastern broccoli industry is the objective of the grant that we have. And I'm going to cover a lot of territory today, so we're not going to go super deep into anything, but really from transcription factors that regulate flower development to transportation models, and I mean semi-trailers when we're talking transportation models. So our goal, this is from the grant, establish an Eastern industry that supplies product year-round. That's one of the market requirements. Let's see, where's my machine? So why would you want to do that? So first of all, there's an opportunity in supplying broccoli. This is per capita consumption of broccoli. Our population is going up and our per capita consumption of broccoli is going up very quickly. It's very, very popular. You read the comments that people put in some of the newspaper articles that are published about broccoli and you think it was poison. But in fact, people eat a lot of the stuff. They want more. Where does broccoli come from? So each dot here is, I believe, 1,000 acres of broccoli production. No, not 1,000. Several hundred acres of broccoli production. But you can see that there's a big concentration over here in California. In fact, in the summertime, it's the coastal valleys where the fog comes in. In the wintertime, there's some production in the Central Valley, but they basically move the operation down to the desert. In the east, it makes a good potato rotation crop. So the coldest, most northern county in the east can produce broccoli. But then it really tails off as you head south, and there's really a very minor amount of production. So why is the opportunity to actually increase production in the east? Very high fuel prices. In fact, typical in the last few years is for California broccoli to sell for $8 in California. It costs $7 to ship it here, and it sells for $15 here, and so the producers here can actually get $15 a box. When you have that big a transportation chunk, there's definitely some opportunity to undercut the competition. Locally grown is huge. The supermarkets are actually asking for it and willing to pay for it. And so supplying locally grown will make it easier for local growers to get in the market than it has been historically when California supply was just a phone call away. Companies are focusing on corporate sustainability. It's a big deal, and they're actually serious about it. And these are some of the companies that you probably wouldn't associate with doing good. They are really paying attention to that. We can shrink the food miles. And in the California production regions, nitrogen restrictions have just come on very, very strictly in the last year. Water restrictions, as some of you probably are well aware, are going to be enormous this coming year. They have no water. So producing an increasing supply in California is going to be more and more challenging. So these, difficult problems in some places, but an opportunity here. Why can't we do it? First and foremost, don't have varieties that are adapted to this climate and can produce reliably. You get unsaleable crop much too often. We also have gaps in production. So Maine obviously is not producing anything this time of year. If you have a gap in the production from an area and go back to another region as the supply source, typically most institutional buyers will continue going to that source. So you lose your customers once a year. That's not a good business model. Fixing that's important. And we also have an incomplete distribution network. It's a lot of truckloads of broccoli, and right now they're heading east and west, and how do you get them to go north and south? You need companies that are in that business and logistics operations, and what are called in the economics business distribution channels. So we have an economic component to deal with that also. Just to give you an idea of what the path is going to be for the next half hour or so, I'm going to talk a little bit about the physiology and genetics of how this inflorescence and bud arrest, that's the important trait, are controlled. Then how we're breeding for wider temperature adaptation, and then talking a little bit about those transport models and what the traits that the market demands. So this is the problem right here. I'm sure you all recognize that as broccoli, and I'm sure you would all walk right past it in the grocery store and not buy it, right? It's okay, tastes fine, yields fine, has zero market value, absolutely zero market value. And this is the most popular variety in the world, grown in the summertime in New York. It's even worse, no market value whatsoever. So this is what happens much too frequently to make a business out of raising broccoli as a primary crop. But it's not that far off, right? It's just a little tweak and you got it. So what's going on here? What's making it bad? So these are flower buds, and the little yellow things in behind are also flower buds. They're just not as big, they're not as developed. And what's happened is that there was a high temperature event earlier in development that resulted in those smaller flower buds stopping development for a little while. So the larger ones continued to develop and continued to enlarge, and the smaller ones sat there and said, well, no, it's not the right time of year, I'm going to wait. And then they started growing again. So they'll look perfectly fine, just not at the same time as the other ones. So the challenge is to get them coordinated. And here's a couple of quick other examples. So cauliflower is basically the same thing as broccoli, it just arrests at a different stage of development. So this is all inflorescence meristem. So each primordium that comes off that meristem makes another meristem that starts producing primordia until you get zillions of them. I don't have the metric number for that, but that is what a cauliflower head is, one big inflorescence meristem. But if it gets too cool, it actually pushes cauliflower towards reproductive behavior, and you get little floral primordia on them. And it starts to look a little rough. So this happens to be a green-colored variety that's unrelated to the development. And it even has little bracts peeking up around the whorls that are going to go. But those little floral primordia make it look bad. You can't sell it. And the technical word for this is ricey, because it looks a little bit like rice. If it gets too warm, it goes towards vegetative. And so you get these little bracteoles forming. And the technical term for that is fuzzy. And it's a good descriptor. And you can't sell that either. So here we have temperature affecting how it develops. Now, you may not have noticed, but making it warmer makes it more vegetative. Making it cooler makes it more reproductive. So developmentally, heat makes it go backwards, but makes it grow faster. So you get faster growth, faster vegetative development when it's warm. So it's a little counterintuitive. So here's a little bit what's going on developmentally and where the sensitive stage is. So this is work I did with Karen Pearson. You'll see the people that actually did most of the work down here in the bottom left-hand corner on most of the slides. But so this is the development of the head and the sort of the key developmental stages that are then regulated by the expression of various homeotic genes. So here's vegetative. You've got primordia coming off. They develop into little leaves. Then you get the reproductive transition. This is what, vernalization, all that, triggers a reproductive meristem. Looks almost exactly the same, except now it's fated to produce reproductive meristems off of that. And you can see there's a little gap there in the by the leaf primordium, and the dome is a little bit bigger. But then you get these reproductive meristems continuing to form off that central one. And it keeps going, gets bigger. You see the little bracts. You can almost imagine a little cauliflower head. And then eventually it switches over. And so there are these little knobs that are bald. Those are floral primordia just starting to form. So this is the second transition here, where those reproductive meristems now become flowers instead. And of course, the flower is terminal. It doesn't develop more meristems. So if it's cauliflower, it continues to do that. If it makes this switch when it's relatively young, it makes broccoli. In a lot of brassicas, you get all of the stages happening at once. But in this one, it's kind of nice because they're very discrete steps. And what we found was that the high temperature exposure somewhere in here causes that difference in developmental stage. So this is the sensitive period where you can expose it to heat. So we've taken advantage of that both for doing some physiology and for doing the breeding. So thinking about what's the genetic basis of the different stages of developmental arrest, how long they stay where they are. So when we started this work, the genes of floral identity were really hot stuff. So that was the place to go looking. So apetala 1 and a gene called cauliflower, because it makes Arabidopsis look like cauliflower when it's in a double mutant with apetala. That those had been described and were really prime candidates. And so a group in England did a controlled cross with a population between cauliflower and broccoli, and they came up with the model that if you have all dominant, you get broccoli. If you have all recessive, you have cauliflower. And if you have some combination of dominant and recessive, you get intermediates. They were using double haploid, so we don't know what happens if it's heterozygous at a locus or not. But this is the basic model. And so if we have something that hinders the expression of these genes, if they're dominant, then you could get that arrest. And so we looked at the collection of all of the broccoli and some of the cauliflower in the germplasm repository and genotyped them all to see how well does the genotype at those two loci predict the phenotype that we see. And so we tried 4 genetics models. This one is that it's all about the cauliflower gene. Second, it's all about the apetala gene. One is that it's dominant by locus. So if you have one locus, or the two loci are dominant, but the alleles are not within a locus. Does that make sense? That's the model. And then the other one is that each dominant allele of either locus, they're all equivalent. It's just how many dominant alleles do you have. So those were our 4 models. And we found that each of those models was significantly supported. So we can't distinguish among them, but those genes have something to do with the phenotype. But then you look at how much of the phenotypic variation was explained by these genes. And not much. So yes, those genes do what they're supposed to, what they do in Arabidopsis, but there's an awful lot more going on. And as some examples, we have here a couple of perfectly good commercial broccolis. They're dominant at the apetala 1 locus. Here's a perfectly respectable cauliflower. It has exactly the same genotype. And here's a perfectly respectable broccoli that's recessive at both loci. So yeah, they do something, but that isn't a very strong case for those 2 genes being the whole story. Just a quick review of what the story in Arabidopsis is, much simplified. But as you go through development, there's a vegetative meristem, then there's that transition to the inflorescence meristem, the transition to floral primordia from meristem, and then you have a bud completely formed, and then there's anthesis when the flower opens. So that's just the development. And so the homeotic genes that control this, you have the terminal flower, which kind of maintains the meristem going. It drops off. Then leafy, which of course being named after a mutant does the opposite of what its name is. That drives me crazy, but that's the way it is. So leafy comes in to start the reproductive development here. And then we have the apetala and cauliflower are involved early on. So the question is, does turning on or off one of these guys, is that associated with the arrest? So we did, well, I can show you this. So just looking at, where is leafy turned on, and you see in the vegetative meristem, it's not really there. So leafy expression is the white silver crystals. And it's when you start getting the reproductive meristems, you get high levels of leafy expression, but then in the flower buds themselves, it's rather low. So that actually fits the model pretty nicely. So what we used was this is actually a commercial Taiwanese cauliflower that happens to be green. But it does something nice. We can keep the genotype the same, but vary the phenotype by exposing it to different temperatures and looking at how the gene expression changes. So when it's cool, it makes flower buds, right? Cold temperatures drive reproductive development. Intermediate temperatures, we get floral primordia. And warm temperatures, we get cauliflower. So just a close-up look, cauliflower, right? Everybody agree? The little bumps you can imagine on this one that those are floral primordia. It's just like little lollipops. There's no further development on them. And again, flower buds, perfectly respectable broccoli. So we can get those phenotypes. And so we looked at the relative expression. So this is using RT-PCR so we can compare the expression in one condition versus in another to see whether a change in expression of a gene is associated with a developmental arrest or permitting crossing that developmental change. So here, going from cauliflower to starting the floral primordia, when that step happens, we see apetala 1 is more highly expressed. We're not seeing terminal flower turning off, which the model would have predicted. We're not really seeing leafy turning off either, and cauliflower isn't doing anything either. Then looking at the next step from that floral primordium to a flower bud, that's when we see the leafy dropping off. And the other genes are relatively constant. Terminal flower may be dropping. Wasn't detectable for us, but there's just less tissue that would have terminal flower expressed. So that's a hint that some of the things are happening, but not the change in leafy is happening at the wrong time for that model, the model as we understood it. There's also another very, very closely related homeotic gene, MADS genes. These are all, they bind with each other, and it's the dimers and the tetramers that are actually binding as the regulatory regions of other genes. So it's a great big mix and match game. And so with 4 loci, a lot of possible genotypes here, but we tracked them all, and they're all declining slowly and as the development proceeds. So potentially keeping the expression of full could be a mechanism for maintaining the arrest stage. So here, this is sort of a schematic of how wide it is how strongly expressed it is. And so here's on the right is a summary of the data I just showed you. The main point I want to make with this is that it's the same players, and some are getting turned on and some are getting turned off, but not at the same time as they do in Arabidopsis. So our knowledge of Arabidopsis is helping find some of the culprits, but not giving us good guidance on what they do. So at about the time we did that experiment, they were discovering lots and lots more genes that have control in Arabidopsis and other things that may play into it. And just the RT-PCR approach was not going to work for us for exploring that further. So I did, Lisa Earle and I did like 4 grant proposals together trying to pursue that with some transgenic technology, and we didn't get funded for that, unfortunately. But there's techniques that are available right now that are really, really promising. And John Hua, who's here, we're trying to cook up something where we can use the GBS to really look at a full survey of what's going on in the various things we've already phenotyped. I mean, it's just sitting there. So I'm really looking forward to where this is going to go. There's a lot of promise there. I'm going to talk a little bit about the breeding now. So when I was a relatively new student here, I had not taken any plant breeding whatsoever. The paradigm I understood was plant physiologists discovered really cool stuff, published it in papers, and then the hungry but ignorant breeders would discover those papers and say, aha, I shall breed for this thing. So there was this young, relatively new professor who was lenient enough to let me into his advanced plant breeding class, where I promptly learned that plant breeders, by and large, don't care whatsoever what plant physiologists do. They keep discovering all kinds of irrelevant stuff. And so it turns out that we have some plant breeders working on this problem, colleagues actually in constant communication with them. But so they used some of our screening technique, but also knew some art and made some good progress. So that for breeding, the problem with this had been huge number of escapes when you try to do just selection for ones that look good. There are many reasons why they could stay looking good. Basically, the heritability was horrible, and nobody was making any meaningful progress with the problem, and it just wasn't a high priority enough for any breeding program to really put sufficient effort in it to overcome the problem. But the knowledge that there's this particular time when they need to be exposed to heat turned out to be helpful in the breeding. And so Philip Griffiths in Geneva, in fact, working with material that Mike Dixon, his predecessor, had worked on, and the guy who got me into this in the first place. And also Mark Farnham, who's at the USDA Vegetable Lab in South Carolina. They had both made really nice progress. And so here's a popular variety in August. No good. This is one from Mark Farnham. That's starting to look like a real broccoli. You could sell that. It's not perfect, but you could sell that. And so the SCRI was passed in the last Farm Bill, and they said, you know, with this, we want you to find a really big problem and find a complete solution to it. And I think everybody said, oh my God, I can't do that. I certainly did. But we ended up putting together a project that got funded because it covered so many bases. And so we wanted varieties that would tolerate the eastern environment. So what's special about the eastern environment here compared to where it's typically grown? Our nights are warm. That's the problem with the different flower bud sizes. It rains a lot. It rains on the harvestable part. That makes it rot and other unpleasant things. We got lots of bugs. We've got lots of diseases. And we don't have a great deal of specialized harvest labor. One of the big broccoli farms in California, I was talking with them about potentially bringing some of their labor here to train people in picking broccoli. And he was saying, oh yeah, that's absolutely essential. There's no way we would have like a lettuce harvester or cauliflower harvester harvest our broccoli. They just couldn't deliver the quality we need. So if that's your standards, that's what I mean by specialized harvest labor. So here's our gaps. So Maine can do great in the middle of summer if you're looking at the course of the year across the bottom axis. And so in the south, you can get harvest around New Year's. And there's a little window in the spring. The middle of summer is fairly challenging. New York can do well in like September into early October. But these gaps in between where you lose the customers. And so we want to increase each of those by a little bit. We're not trying to have broccoli grow everywhere all the time. We just need it to have the different parts of the East Coast overlap enough. So we need just a little bit of improvement, like that first slide. It's close, but not going to cut it. So we need just a little improvement. So the grant covers breeding, regional trialing of promising breeding lines with feedback to the breeders, releasing and selling varieties. This is a really big deal. And seed companies are really good at that. And so that's their function in this. They're also good at breeding, for that matter. Establishing a grower base. I'm not going to talk about it here. We have a substantial extension objective because the growers have to be really good. The competition is excellent, like that grower who said he wouldn't have a cauliflower harvester pick his broccoli. They need to be that good to get into the game. We need to have a distribution network that makes sense to the growers, to the distributors, economically, and to the supermarkets. And consumer acceptance. If there's a premium for local, or if they look a little bit different, we need to find out that people will want it, and also feedback to the breeding if there are traits that the consumers are reacting to, either as positive signs of localness in some physical attribute, or something we thought didn't matter too much, and it turns out it does. So that's among the traits coming in. So we have 3 public breeding programs and 3 private breeding programs are involved with the project. So my observation, the physiologist, every breeder has a unique breeding philosophy. Even if you're breeding for the exact same trait, they all think about the problem in their own individual way. And that's great. That's a lot of fun for me. But it means I'm not going to tell you anything about the nitty-gritty of how they actually do their selection, because that remains an art and a mystery. They all have distinctive breeding populations, at least to some extent. The practice in broccoli, it turns out, is you take the best-selling variety and you self it back and start crossing it with everything you've got. That has been a fairly common technique among breeders of broccoli. So to some extent, they've all stolen from each other. But they all have distinct breeding populations that they're working with also. So to bring a new trait, which is really what we're doing here, something that may be kicking around in their breeding population, but they really hadn't been making a big effort to select for it. Some of them are going to hit the jackpot, some are not. You don't know until you try. Encouraging competition. So my job as sort of the ringmaster in here is to egg them on and say, look, here's what the group did here. Don't you have another cross you want to test? Don't you think you need to look over at some of these potential parents? Just see that they are putting as much effort into this as they can, because they all have a zillion distractions. So the competition is actually fun. And also the licensing of public material. Obviously, the public breeders are not going to be releasing and selling varieties. That's not their business. That's the seed company's business. So we want the seed companies to buy the public material. So there's also a little bit of salesmanship going on in there. So these are the breeders. So Mark Farnham's the one who really kicked this going, but Griff has a lot of good material, and when he starts moving on something, he starts moving on something. Jim Myers has been breeding broccoli for a long time in Oregon, and he's got 2 traits that are of interest. One is that they have this exerted head that comes up above the rest of the plant, so you can see that it's ready to harvest and it's easy to reach it. Originally, the intention was mechanical harvest, but we're not there yet. And the other is that he's leading the Novik project, which has a lot of fingers here with Michael Mazurik in particular for breeding organic broccoli. And so he's been taking charge of making sure there's an organic specific broccoli being developed. But then we have Seminous vegetable seed. That's a division of Monsanto, which I get a lot of comment on. Case Sintony is the breeder with Bayo, so it's a Dutch, privately held but multinational company. And Syngenta, again, their breeding has just recently moved back to Holland. But John Hewitt, who was a postdoc here in this department a while back, is their US contact. So what are the traits we're looking at? So the bead uniformity, the main thing I've been talking about. So having a low requirement for the chilling hours, if you want to call it that. Having a good dome. So A dome sheds rain, so the water runs off so it doesn't rot. It's that simple. High temperature tends to make it flatten out, so we need an especially good dome on these. We need good head uniformity. People are persnickety about the lumpiness of the head. So the extension trait, Meyer's exerted head for easy harvest, is a valuable trait. We need to have normal or higher carotenoids so that broccoli is good for you. People eat broccoli because it's good for you, right? So ours better be good for you as well. And normal or higher glucosinolates. This is being done by Carl Sams and Dean Kopsel at the University of Tennessee. You need the head to be firm because the supermarket buyers really expect that. You need small flower buds. This is really a pain because it's a hard trait to get in there. But that makes it look good, at least to Asian wholesale buyers. We don't know if Eastern consumers care. So that's why the caveat is here. The West Coast is growing for the Asian eye. It needs to be really green so it looks good for you. People associate that color with the nutritional qualities, absolutely. And you saw the green cauliflower. They come in all colors. Purple, red, pale, blue, all kinds of things. It has to be fecund. The seed companies are in the seed business after all. If they don't produce a lot of seed, it's not a product. And we need stability across a lot of environments. They need to be widely adapted. So we're doing these ratings in multiple locations. We've got rating cards, so everybody rates exactly the same way. So we have genotype by environment interaction, not genotype by rater interaction. That is a significant problem. We spend a lot of money getting people together rating the same way. So bead size, you have it in the field, you get that. What does the dome look like? How uniform is the bead? We need a lot of examples because it looks ugly in many different ways. And how green is the green? Ignoring the purple, just the green. It's not quite as simple as it sounds, but everybody's rating the same way. So our trials run through basically 3 steps. We've got the same 9 traits that we're rating in all of phase 1. But so phase 1 is looking at new crosses. This is where all of the breeders are trying new stuff, either from their own programs or between programs. Again, each unique personality decides that it's ready for this phase at a different stage of development. But we do a bunch of entries, 5 locations, a couple of plantings. We try to do a stressed and an unstressed. What can it do in good conditions, and how much does it suffer? That wide adaptability. Some of these do quite well. They go into phase 2. We have fewer entries there, but we do 5 different plantings at each location. So we have 20 plantings each year out of that phase. So we really have a lot of environments to compare quickly. And so we can assess that stability. And this is where we also do a nutritional evaluation on everything. So, first and foremost, we're going to throw out anything that doesn't cut it nutritionally. That's the really simple use of that. But it gives us an opportunity to look at the genetic and genotype by environment impacts on the nutritional components, which people really care quite a bit about. Then phase 3 is basically grower trials. Yield, how many cuts does it take to get all of the crop? Things that growers are very concerned about, but our research farm trials are not going to tell them anything about. So that's basically what we do. Here are the locations of the trials and the people who do them. So we go from southern Maine to southern Florida. They're all planted at different times. So we have the coast where the Atlantic is an important moderating influence. Long Island has a lot of potential. And then we have down here the mountains. So it turns out in the Asheville area in North Carolina, they have a shot at harvesting in July. They'd have the entire southeast market to themselves for local broccoli if they were to expand that. And we have a lot of potential for acreage in western New York. So here's one way to look at stability. I'm just going to show you some of the controls here. We do this on all the test varieties also. But if you look at the rating, so all of our ratings are 3 is marginal commercial quality, 4 is spot-on commercial quality, and 5 is spectacular. That's the 5-point rating scale, but the part we're interested in. So the median location, so this is how they did in 28 different plantings. So the median rating was just about 3 for all of them. Not really any difference. At the best sites, we had 2 that started looking good and 2 that still looked pretty ho-hum. But here's the thing that the growers worry about. So if you look at the 20th percentile, so 1 year in 5 or 1 crop in 5, it's going to be this bad. We have 1, still marketable. We have a couple, not even close. So that's the kind of stability we need to see in a really good variety. So looking at how often does it do awful is an important parameter there. And here's some of the things, the kinds of data we pull out and are thinking about it. Here's just looking at a principal components analysis of all the traits that we measure. First of all, you can see that the data spread out very nicely, as they should. We don't have any clusters there. But we find that 2 of the traits that are loaded in the same direction are bead size and firmness. It turns out that small flower buds pack more tightly, and big flower buds have air inside them, and they squish a little bit. So that those 2 move together turns out to be unsurprising. And over here, we have a couple that we really care about, the dome and the bead uniformity. Those are the traits that the heat really affect. And they're moving together. Yahoo, that's great. That was completely unexpected, but it may be easy to select those together. And the other thing we worried about is whether the small beads don't hide any of the imperfections because they're too small. If that were the case, that would be aiming in the opposite direction of the bead uniformity, but it turns out they're perpendicular to each other here, so there's the potential to select them independently. So we do a bunch of these biplots, just where you plot all of your traits against all of your traits in their rating, and I'm going to zoom in here a little bit. So some of them, there's a strong positive association. So firmness and bead size, like I just said, they tend to go together. So they're positively associated, right? That makes sense. Some of them, there's no relationship. So plot uniformity, plant to plant uniformity has no correlation with firmness. That makes sense. And that's true of a lot of them. But the interesting ones are here. You have a negative correlation between 2 desirable traits. So the upper right-hand corner is desirable in all of these associations. And so bead uniformity we really want, bead size we really want. So this is the one where they were perpendicular in the principal components analysis. So they're negatively correlated, but not super tight. And so for a physiologist, I look at this, okay, they're negatively correlated. See, a plant breeder doesn't look at it that way at all. They look at, oh, I want the stuff in the upper right-hand corner. Let's ignore all those other data points and keep those, right? We just called those out. That's the cultural difference here. So great, they're not tightly correlated. You can select some good ones. And we actually do have some nice ones. And so what we've looked at, this is a way to assess how well adapted they are to stress. So we just do a spider plot of certain traits. And the green line is how they perform when conditions are good, and the red line is the stressed condition. And you find something like here is Ironman, for instance. So it looks pretty good. It's getting high scores all the way around when conditions are nice, and then it really collapses for several of those traits when there's stress. But then we've got some new material on the bottom row where there's really very little difference between the red line and the blue and the green line. So the temperature or the stressed environment, as we understand it anyway, isn't having much effect. It's getting mediocre scores for firmness and bead size, which just means it has big flower buds. Again, we're not convinced that the Eastern consumer is going to be worried about that one. So this is one that would definitely move forward. We also find some instances like number 2 is a good case, where we're getting a small bead. So firmness and bead size are the first two here as you're going around clockwise. And we're finding that together with something that has good bead uniformity, good dome, and neither bead uniformity nor dome moves at all with stress. So we're combining some different traits that we thought would be difficult to combine. They're showing up together. This is very promising. So the seed companies so far, after 3 years of breeding, the seed companies have released 6 varieties. We're expecting 2 new varieties to come out. So this is stuff that was in their pipeline largely. They're now making crosses between public and private programs, several crosses among the public programs to really put together the traits we want. So we're now actually in the field with varieties that are an incremental improvement over what we had. And yeah, so there will be a lot more in the pipeline. So the breeding is very exciting, coming right along and going all the way to the seed bag and the seed catalog. Give you an idea of the transportation, because this is something we don't often think about. Say, oh, we'll just plant more. And this is the flow of broccoli in the summertime when most of our production happens. So you have sources in California, source in Maine. And so all of this California broccoli is coming in. The circles are the major markets. And so this is something Shadi Atala, a student with Miguel Gomez in the Dyson School, have worked on. That Maine comes in and basically hits New England and New York City. And increasing production in upstate New York would generally supply markets in western New York and west of us. We would have a lot of trouble getting into the New York City market because of the supply from Maine. Wouldn't have expected that, but that shows you where to target the efforts. And the Carolinas, rather than going north to the DC, Philadelphia area, makes a lot more sense to head south towards Atlanta. But so our model shows, this is on the sustainability, if Eastern production went up by 30%, and that's not a whole lot, the total US cost of broccoli, so the amount of money spent on broccoli in the United States drops by 1%. That's not bad. I mean, 30% more of a tiny production area having that big an impact is not bad. It also reduces the food miles of the average Eastern broccoli by 100 miles. That's not bad. That's a significant effect on food miles. It's still around 1,500, but still, it takes it down some. So we can hit the trait, if you will, of sustainability a little bit. So the consumers, they care about appearance, potentially the tenderness and sweetness of local. So Xiaoli Fan is a graduate student also with Miguel, and she's been doing the consumer trials. So here is a set of consumers we did in this wonderful laboratory in the newly renovated Warren Hall, where we can do testing. So we actually excluded students, only real consumers. So I had to go through IRB and everything to be allowed to do this. But basically they got 3 samples, so 3 that they got to taste that had been parboiled, and 3 heads where we had the control. This was a California broccoli in the middle. Now basically looks perfect. Then we had A was a breeding line. It's hard to tell in this picture. It had the larger flower buds, and because of the wax on the surface, it also came across as a lighter green color. Just a little bit. Now, we all thought it looked great, but was that qualitative difference meaningful to the consumer? It's really important to feed that information back to the breeders. And C was a good commercial variety, but it was just showing a little bit of heat stress. So how tolerant is the Eastern consumer of this primary appearance defect that we get? How much can we get away with? How much do we actually have to improve that trait? And how much can we tolerate a lower standard than the Western shippers have? And so it turns out that if you tell the consumers that it's New York grown, then but the middle one is California grown, then they're willing to pay more for the local. And they told us they tasted better. So that had something to do with it. If we just told them it was Eastern grown, so the local connection, the community connection is much weaker with the term Eastern than New York. It dropped a little bit, surprisingly not very much less. So the idea of having the regionally produced product looked promising. If you didn't tell them anything and just showed them the material, then it was a little bit lower for the two new ones. So combining the local with the slightly different quality seems to work. And people read all kinds of things into it. I talked with one of the panelists as they were leaving, and he said, "Oh, I like C the best." That was a little raggedy looking one. I said, "Yeah, that was the organic one. I like the organic one." Nobody had said organic during the whole thing. It was never raised or hinted at. So what went through people's minds when they put these prices on them can be a whole slew of things, but we don't care about the mechanism at this point. We just care about the result. Does it sell? Will supermarkets start buying Eastern product even if it has some little different appearance? And numbers like this says yes, they should. So to wrap it up, we have, so we need to close all of these economic bottlenecks. The seed companies, the growers, the supermarkets, they all have to make money, and they are. So having our breeders cooperate with each other to some extent and egg each other on is working. We're really concerned about the expectations of the buyers. The whole issue with appearance, it's all in buyer expectations. We're really dealing with that. And the number that we put in the grant proposal that just scared me to no end when we put it in, it looks like we're on target that by the next ag census after the project is done should show $100 million of Eastern broccoli production. So I have a slide with a lot of names on it, but I'll just show this one as some of the key cooperators and what I've talked about today. So the one in bold, they gave us $3 million. I'm very thankful to them. Bunch of companies that have agreed to invest an additional $3 million. I'm very thankful to them. So we have Jill Eccleston as my program manager. So we have the breeders that I mentioned earlier, and my colleagues at the Germplasm Repository in Geneva did a lot of the genetics work with me. And Denise Duclos is a fabulous graduate student who did all the gene expression work. So with that, thank you very much.